Hello, welcome to the second installment of Single Cells 2020. My name is Heather and I'm joining you today from beautiful Awabakal country here in Newcastle, New South Wales. I'd like to begin by acknowledging the traditional custodians of this land, and I pay my respects to their elders past, present and emerging. I also extend that respect to the traditional owners of the lands from which you join us, and I pay my respects to all Aboriginal and Torres Strait Islander peoples joining us today. I'd also like to extend a very warm welcome to anyone joining us for the first time today. Oz Single Cells is an energetic and creative community of Australian scientists, and we're always happy to meet new friends. Today we'll be exploring how single cell analysis technologies might help cancer patients. We have some fantastic speakers lined up for you, whom I'll introduce shortly. Each episode in this series will bring you leading science from Australian and international research. We'll also include discussions about the future directions of single cell science and some of the more controversial topics in this field. Of course, none of this would be possible without the generous and continued support of our industry sponsors. So let me say a huge thank you on behalf of the entire community. Throughout this podcast series, there'll be several opportunities for you to participate. You can join the discussion live each week and submit questions for our speakers. You'll also have the chance to compete in some fun challenges and present your own work at our online poster session. If you can't join us live for an episode, you'll be able to view the recording later through your registration link. And on that note, if you missed the fascinating discussion from last week, I strongly encourage you to take a look as soon as you get the chance. Next week, we'll be learning more about how single-cell sequencing can be linked to the spatial organisation of a tissue. So don't forget to register when you receive the reminder email. But without further ado, let me introduce today's speakers. First of all, we have Associate Professor Nick Nabhan joining us from Texas, where he works at the MD Anderson Cancer Center. Nick is one of the founding fathers of single-cell sequencing, being the first person to perform genome analysis in single cells. He continues to lead research in the genetic diversity and evolution in cancer, as he'll be sharing with us today. Hi, Nick. Thanks for joining us.
Hi, nice to be here today. Thank you very much for the invitation.
Next, we have Dr. Vanessa Chin. Vanessa is a practicing medical oncologist based at the Kinghorn Cancer Centre in Sydney. Her clinical interests and also her postdoctoral research are focused on lung, head and neck cancers. Welcome, Vanessa.
Good morning, everyone. Thank you for having me.
Finally, we have Dr. Karis Tay, who is a research fellow at the Walter and Eliza Hall Institute in Melbourne. Her research is focused on apoptosis in cancer, or as she very eloquently puts it, the kill switch that triggers cell suicide in cancer. Welcome, Karis.
Hi, thanks for having me. It's lovely to be here.
I'd also like to introduce Dr. Helen Maguire, who is my co-host today. Helen is based at the Ramaswamy Facility for Human Systems Biology in Sydney, where she coordinates a research program in cancer immunology. Hi, Helen.
Hi there. Really great to be a part of this exciting program. Thanks.
Today's episode is supported by our friends at BD and BGI, so a special thanks goes out to them today as well. This session will be about 1.5 hours, with presentations from each of our speakers, followed by a couple of questions. At the end of the session, we'll also open up for a more general discussion about the future of single-cell analysis in the cancer clinic. So make sure you stick around to the very end for that. If you have any questions for our speakers, you can send them through the Q&A panel in the top right of your screen. Please include your name and location so we can address your question. Also, please engage on Twitter with the hashtags you see on your screen. Just refrain from sharing the slides from today's presentations, as these include some confidential information. So, to get us underway, let me just remind everyone that cancer is a very heterogeneous disease, including many different cell types: tumour cells, immune cells, stromal cells, nerve cells, and blood vessels. In addition, cancers can contain distinct genetic subclones, as Nick will explain further. And cancer cells can also transition between different cell states. Furthermore, this heterogeneity is not stable, but changes over time as the cancer evolves, responds to therapy, or progresses. Of course, this heterogeneity is really important for the clinical management of cancer. The ability of cancers to evade therapy, develop resistance, and spread around the body are major challenges for cancer clinicians. If we can identify and somehow eliminate the cells responsible for processes like metastasis, we will see greatly improved outcomes for cancer patients. All of this means that cancer is an obvious target for clinical applications of single-cell analysis. In fact, routine clinical practice already relies on many traditional methods that provide single-cell information. For example, pathologists routinely grade cancers based on immunohistochemistry. But this analysis is not always straightforward, with variability in staining observed within cancer specimens. Similarly, methods like FISH, flow cytometry, and karyotyping can also reveal cancer heterogeneity. So how can newer, high-resolution single-cell technologies complement these traditional methods and bring benefits to patients? To start our discussion, I'll now hand over to our first speaker, Nick. Nick, could you please check that your microphone is off mute?
Oh, hello, everyone. Can you hear me okay now?
Thank you.
Okay, fantastic. All right, I'll go ahead and get started. Thank you for inviting me here to speak today. I'm going to talk about clinical applications of single-cell genomics, which is an area of great interest to us. So for some time now, we've been interested in how a single somatic cell in breast cancer that would be an epithelial cell is able to acquire mutations and then expand and diverge into distinct lineages, resulting in this big mess of genetic profiles that we call intratumor heterogeneity. And a major question that we've been interested in is whether this genetic diversity is just stochastic, or alternatively, whether it might play an important role in different biological processes in the tumor, such as initiation and invasion in premalignancies, metastatic dissemination, or therapeutic resistance. So for sequencing technologies, there are really three approaches that have been used in the last decade or so to understand intratumor heterogeneity. The first approach involves deeply sequencing a tumor and then trying to cluster mutation frequencies and normalizing by copy number and purity to estimate how many clones are present. And this gives a good broad view, I think, of the major populations that are present. The second approach, which was really pioneered by Charlie Swanton and others, involves spatially macrodissecting different areas of the tumor and then comparing the mutations in those areas to reconstruct evolution and intratumor heterogeneity. This works quite well when the clones are spatially segregated, but it doesn't always work that well when they're intermixed in the same areas. And then the methods that we've been developing and applying are single-cell sequencing techniques. And here we make a nuclear suspension out of a tumor. We profile individual cells, and we compare their genomes. And this allows us to completely resolve the kind of admixture effects of intratumor heterogeneity, and we can then get genetic mutations on individual cells and compare them to understand tumor evolution. So we've been really developers of single-cell DNA sequencing technologies. Some time ago now, we reported one of the first single-cell DNA sequencing methods, and that was really to look at copy number aberrations in tumors. Following that, we wanted to look more at mutations at base pair resolution. So we developed methods to do single nucleus exome sequencing and single cell genome sequencing. We quickly realized that that's quite expensive to do because Sequencing just one cell is like sequencing a human genome or human exome, and you can only do this on a few dozen cells at a reasonable cost. So more lately, we've been focusing on much more targeted assays to look at mutations, so we can profile thousands or tens of thousands of cells. And we've also more recently developed fragmentation-based chemistries to really scale up single-cell DNA sequencing and make it cost-effective. So what are some of the applications of single-cell DNA sequencing in cancer research? They can be used to resolve clonal substructure and really understand what populations are present in the tumor and what kind of mutations do they harbor. They can also be used to reconstruct clonal lineages, understand the timing of mutations or copy number events over time as the tumor progresses. These methods can also be used to look at clonal dynamics and understand in response to therapy or comparing primary metastatic tumors or premalignancies and invasive tumors, which clones are changing, what does selection look like during that process. And finally, of important clinical relevance is the co-occurrence of mutations. So with single-cell data, you can get very precise information on which mutations are mutually exclusive in different populations in the tumor and which mutations coincide in the exact same cells, which is important for targeted therapy. But tumor cells are just half the story. Of course, as a tumor progresses or becomes resistant, there's always the microenvironment, which is co-evolving, and that includes both the stromal microenvironment as well as the immune microenvironment. And so here's just an example of DCIS breast cancer, where we evolved from a very highly differentiated ductal lobular structure embedded in the fibroblasts and adipocytes to a premalignancy where the tumor cells are still within that myoepithelial basement membrane, but then progressing to invasive cancer where everything breaks loose and you get invasion across large areas of the tissue sections. And then there's also single-cell RNA sequencing techniques. And these have turned out to be great ways to do an unbiased profiling of not just the tumor cell expression programs, but the whole microenvironment. And in addition to identifying the different cell types, you can then take a specific cell type. So in this example, we're showing T cells, and you can recluster that data to identify different cell states, which sometimes imply different biological functions, like in the case of T cells. And you can really do this for any cell type. The tumor cells, you can also grab those cells in this cluster and cluster the data to identify differences in various phenotypes like proliferation or EMT or hypoxia. You can also use this data to really understand what do cancer subtypes mean. Is there one dominant subtype in all the cells, or do you see mixtures of different cell types in individual patients? In some cancers, you can also reconstruct lineages of development. And you can also do cell type specific differential expression analysis, which is important because that allows you to compare just one cell type before and after therapy. The field of single cell genomics has really grown a lot over the last decade or so, and I think it's just continuing to grow. There's over 800 publications, I would say, now in 2020. It's expected to reach a $4 billion market. And the areas that have been impacted most are still, I think, cancer research. That's where most of the publications are. But there's a lot of work being done in microbiology, neuroscience, prenatal genetic diagnosis, immunology, and many other fields. And so there's different technologies available, as many of you know. There are techniques to do single-cell DNA sequencing. This includes both mutations and copy number alterations. There are approaches to do single-cell RNA sequencing, now using nanowells or microdroplet approaches, and methods to do single-cell epigenomics, which most commonly involves things like ATAC sequencing to look at chromatin accessibility, but also methods to do DNA methylation and look at chromatin interactions as well. Now, the future is kind of combining these methods and being able to, in the same cell, get both DNA and RNA information or RNA and epigenomic information. And a lot of those techniques, I think, will be forthcoming in the next year or two, especially in higher throughput formats. Now, if we take a look in cancer research, we'll see that most of the initial studies kind of focused on either looking at intratumor heterogeneity in the primary tumors or looking at the microenvironment with single-cell RNA sequencing. Following a lot of that initial work, which of course continues today, more studies were comparing circulating tumor cells, especially papers from Dan Haber's group at MGH, and looking at metastatic dissemination. And I think two new areas that are very exciting to me that we work on and others do as well are premalignancies, where the spatial context is really important, And also therapeutic resistance, where you're dealing with things like core biopsies and fine needle aspirates and very limited numbers of tumor cells. So thinking now about how we can do single-cell sequencing in the clinic, we have to really consider a lot of things, especially the sample types and quality. Some single-cell sequencing assays, like single-cell RNA sequencing, really require fresh tissues or cryo-stored viable cells. And so that presents a big challenge to pathology suites and just to pathologists and surgeons in general, because the samples have to be processed in a very quick time frame. Now, there's some experience that people have had in hospitals working with xenografts or organoids to do these types of things, but still, it's a big effort if you really want to do single-cell RNA sequencing. Other methods like single-cell DNA sequencing, of course, can be compatible with most types of samples, whether they're snap frozen or viable or anything else. And then, of course, new methods that we and others are developing for single nucleus DNA sequencing can be used on FFPE tissues. And that's very exciting because you can have long-term clinical outcome data to tie to that. And there are also methods like single nucleus RNA sequencing that some groups are starting to use on frozen tissue samples. I think there's still open questions about how good the data quality really is, but at least there have been some demonstrations of doing this. And then the other thing, of course, is setting up really the protocol to have these tissues processed. Will there be FACS sorting or not? Will the cells be cryo-stored, or will it be run directly after the surgery? So a lot of considerations, I think, for setting this up in the clinic, which we can discuss in more detail later. Just for one example, this is our protocol for the Human Breast Atlas project. And so in this protocol, we consent the patients, one of the surgeons or one of the clinicians will do that. And then we'll have different sample types. After the surgery, we have somebody pick it up and jog it back to the lab after pathology in about an hour or two, dissociate the sample, run the single-cell sequencing experiments, and then often cryo-store some of the cells as well. But the whole process takes quite a long time, and it's not really that easy to set up initially, just because of all the logistical complexities. Now, in terms of applications in the clinic that we are excited about, and I think others as well, we think there's real opportunities in early detection, not just in the blood for looking at circulating tumor cells, but in many bodily fluids. For example, the cerebral spinal fluid to look for tumor cells, to look in the urine for early bladder cancer cells, look in the spit or the sputum for early lung cancer cells. And the beauty of having single-cell methods is if you can identify them, then you can pick out those cells and get a very pure signal for measuring the genetics or transcriptomics. Risk stratification is another area where we think these methods can really help to determine which patients are at more risk for progression, like in pre-malignant cancers. These methods can also help, we think, with matching the right patient to the right trial based on the microenvironment as well as the tumor cells, predicting therapeutic response, and then really drug discovery applications. So can we identify new drug targets in the microenvironment with these tools? Same goes for the tumor cells. We can reconstruct lineages and look at clonal substructure and determine whether we should target truncal mutations, which might be present in all the tumor cells, or can we just identify populations that we think are the most malignant and target those to prevent metastasis or resistance. And then non-invasive monitoring of circulating tumor cells. There are already a lot of great methods to enumerate CTCs in response to therapy. And now with these tools, we can actually pick them out and profile them to understand if there are mutations that might be more informative for which therapies to select. And I think it's a little bit idealistic, but one day what we would hope is to be able to take a tumor like this, perform single-cell RNA sequencing, single-cell attack sequencing, and then really identify which cell types in the microenvironment are reprogrammed, and then think about specific targeted therapies. There are some examples here for targeting just those cells in order to really treat the disease, or even multiple cell types based on these inhibitors. So now I'm just going to give a few quick examples of some research studies that we have done where we think there are clinical applications, although these studies are still really on the research side, but we can talk a little bit here about how these could be extended into clinical developments. So the first is on chemoresistance and triple-negative breast cancer. This involves a postdoc of mine, Ruli Gao, who's a faculty now, Carissa Kim, who's an MD PhD student. She's a resident now. and a collaborator at Karolinska Hospital in Sweden that we worked on for this project. And this was focused on triple-negative breast cancer, where patients, about 50% of patients do very well. They actually have good long-term outcome, but 50% develop resistance very quickly within 1 to 2 years, develop metastatic disease, and have very poor outcome. And the mechanisms of resistance are not well understood. And with single-cell DNA sequencing methods here, we were able to profile cells from core biopsy samples and compare them before, midpoint, and after treatment. And this was to look at the effect of chemotherapy. And in some patients, as we can see here in these single-cell copy number profiles, there are about 3 major clones present. When the patient received chemotherapy, they were eliminated. So after the final time point, we couldn't detect any single cells that were present. And so this is a PCR patient that responded very well. And we can really track this in a lot of detail using these single-cell methods. Here's another patient. So this is a patient that did not achieve pathological complete response. They had 3 clones present as well. And over the course of treatment, what you can see happening is that a small population that was around 7% initially really expanded to around 71% and constituted most of the resistant tumor mass in this patient. And if you compare the genotypes of these 3 clones, you can see that the clone that was resistant had some very specific aberrations, including on chromosome 3p, these 2 deletions of RARB and also of IL5RA. And so we think this is a way we can potentially track and monitor the resistant clones over therapy and understand clonal dynamics as patients are developing resistance. And so we saw these 2 responses that we call clonal persistence and clonal extinction. You can also think of them as PCR and non-PCR. They correlate pretty well, but not perfectly. There were some patients that had PCR, but we could still detect some residual tumor cells here. And we can also do gene expression analysis, so pre and post treatment. And when we did this in these patients, we found that there were a few signatures that were upregulated in the resistant disease. AKT1 survival signaling, hypoxia, EMT, angiogenesis, and degradation of the ECM. So this has helped us to think about possibly new ways we could target these patients once they become resistant, hopefully to overcome the resistance. And the second story I'll tell you about very quickly here is on invasion in pre-malignant breast cancer. And this involved a graduate student, Anna Cassasen, who's a postdoc now, Mary Edgerton in pathology, and Aislinn Schalk, who's a quantitative student in my group. And DCIS is a pre-malignant breast cancer, and about 10% of low grade will progress to high risk, invasive disease. About 30% of high grade will progress to invasive disease. And so it's a big clinical problem, understanding which of these tumors are likely to progress and which are not. In this study, spatial context was really important, and I think you'll hear more about that at the show next week. But we had to develop a method here that could preserve the spatial information of single cells while we profile them genomically. And so we call this method topographic single cell sequencing. It involves combining both imaging as well as laser capture microdissection and a technique called laser catapulting to catapult cells off the slide, collect them, amplify their DNA, and then do multiplex barcoded sequencing, and then tying the imaging data and the spatial coordinates to where those cells were located in the tissue. Here's some data from one patient. So this is a clustering of single cells. And you can see there were 4 main clusters. One was just diploid, normal cells, and then 3 tumor clones labeled as A, B, and C. When we compare those 3 tumor clones, we found that they share a lot of genetic mutations, including amplification of MYC, but they also have very distinct aberrations too, including amplification of MET, amplification of EGFR that was only in the C population. And importantly, we found that all of these clones diverged very early on within the ducts prior to the invasive event. And as they invaded the surrounding tissues, it really involved just the same genotypes spreading out in the tissues rather than new mutations or copy number alterations being acquired during this process. And so we could map these spatially to different areas in DCIS tissues. And it was very interesting because some of the clones were much more localized to the ducts, like the black one here, clone A. But there are some cells spread out in the tissue, just very few. While other clones like clone B were really spread out everywhere. They seemed to be much more ubiquitous throughout the tissue. They'd spread out somewhere in the ducts, but a lot of them were different tissue regions. Which may imply that they were more migratory. But again, this is just a single snapshot in time. So in all patients, what we observed was that the copy number aberrations had occurred much earlier on within the ducts, and then this process of invasion really involved just those same genotypes expanding to form the invasive tumor mass. And we did see some changes in frequencies, which might imply some differences in selection. And so our data, did not support this independent evolution model where it's thought that the in situ invasive disease are completely different genetic lineages started from different cells. It also did not support this idea of an evolutionary bottleneck, which was quite prominent in the field. The idea that you have many different clones initially in the ducts, and then one of those is selected and expands to form the invasive tumor mass. And instead, we saw evidence for what we call multiclonal invasion, where the genetic diversification occurred early on, and then all of the clones co-migrated into the invasive areas to establish the DCIS breast cancers. Okay, and the last part that I want to mention is this project that we're doing called the Human Breast Atlas Project, which is a collaborative project between our group, investigators at UC Irvine, and also Cold Spring Harbor, as well as all the people in my lab that contributed to this project. So this is not a cancer-related project. This is to understand just the normal biology and physiology of the human breast. So the human breast is a network of ducts and lobules embedded in fat and adipose tissue. And there's about 10 cell types that are pretty well known from pathology. And the breast is very important to consider all the biological variables such as parity status, menopausal status, menstrual cycle, etc. This is just an H&E showing you some breast tissue. You can see these different areas. There's some areas that are just all fat adipocytes, some areas that have epithelial cells, ducts, and lobules, and then some areas that are all fibroblasts or connective tissue. So the scope of the project is that we're going to look at 100 women over the next 3 years. We're going to consider all the biological variables, and we'll be doing single-cell RNA sequencing, single-cell attack sequencing, single nuclei RNA sequencing. That's mostly to get the adipocytes, spatial transcriptomics, FISSEQ2, and we'll also be building organoids to do some validation. And this is just the pilot data of the first 20 women or so. You can see the 3 major epithelial populations, the basal epithelial cells, hormone negative secretory luminal cells, and hormone positive luminal cells. You can also see the vascular and lymphatic cells, and surprisingly, a large number of immune cells, which we did not expect going into this project initially, as well as some underappreciated cell types like parasites. But as you can see, there are no adipocytes here, and so that was a big surprise to us because the breast is mostly adipocytes. There's a lot of variation in these cell types across the women, as you can see here, and we still don't know quite how these correlate to various risk factors such as disease status, but we can look at them and try to understand this heterogeneity. And then for every cell type, for example, for the fibroblasts, we can look at what the top genes are that express and what are the markers. And for every cell type, we get about half the markers that are known and have been established in the literature. And then we discover about half of the markers which are new and we think are important to understand and might provide new markers to study these cell types. But then the really cool thing about having a single cell RNA data is you can take that data, you can recluster it and identify cell states. And oftentimes cell states correlate to different biological functions. So we're here with the fibroblasts. We had these 5 different cell states. We had fibroblasts that looked like they were involved in matrix remodeling, some that were associated with adipocytes, and so we call these lipofibroblasts, some that look like they're activated, express things like FAP and other markers that might be even pre-malignant. We have this FBN1 subtype, and we have a myofibroblast population as well. So, I think that's the end of my slides. I just wanted to leave it open for some discussion questions, which we could talk about now or later. One is, how will we really implement single-cell genomics in the clinic, provided the many logistical and technical challenges working with pathology? Can single-cell sequencing really be used directly as a clinical tool, or is it more of a discovery tool where we can identify important drug targets, or we can understand biology, and then we might use a different clinical assay that's more targeted later on. Wanted to stress the importance of establishing these normal references of human organ systems. Otherwise, we just can't understand diseases like cancer. And an open question, I think, is just how well can we get these tools to work with FFPE tissues, which of course is a major resource in those clinical environments. And also, I think it'd be fun to think about, which are the most immediate clinical applications that we can realize, in a few years, not in 10 years or 20 years, but, somewhere in the near future. And that's really the end of my talk. Just want to thank all the people in my lab who contributed to these projects, our collaborators here at MD Anderson and other places, as well as our funding support. So thank you very much.
Thanks, Nick. That was fantastic. And I do note that you've got some postdoc positions open. So if that got your interest, yes. Definitely get in touch with Nick. Not a bad plug. So we've got a couple of questions, and I encourage the audience to add some more if you'd like. I'll start off. We've got one from Christine Wells, who's really been captured by the depth of information that you can. He's been really excited by the depth of information that you're able to get from these tumor biopsies. So in that idea, what we learn from the cancer, how is it going to help us with progressing treatment? How do you see this going from describing the problem to predicting the outcome to the patient? It's kind of a big question, isn't it?
Yeah, absolutely. So I think for risk stratification and predicting patient outcome, the challenge is that you usually need long-term clinical information. And so that's, of course, a problem when you're collecting fresh, viable cells, because you still have to wait another 5 years or 10 years to know how that patient really did long-term. So I think at least with risk stratification, probably we'll have more clinical answers sooner with things like DNA assays or using frozen tissues. Single nucleus RNA sequencing might be an option there because those samples can be archived, although the longer they're archived, probably the poorer the RNA quality. So yeah, it's a challenge. I mean, some projects that we're doing now, for example, in DCIS, using single-cell RNA sequencing, we're not going to know for a long time really how those patients did. And so that's also motivated us a lot to work on FFPE materials and really kind of get single-cell DNA sequencing methods working in those types of tissues and archival materials so that we can answer some of these more clinical questions where we need outcome data.
Yeah, great. Yeah, I mean, it's a balancing act, right? So you've got material available, or do you go ahead and design something that needs new material? Then, that's tough. Okay, so we've got another question from Anna Trigos, who has asked for you to comment on the advantages of single-cell DNA sequencing compared to deep bulk sequencing. So I guess she's speaking in terms of the number of clones and detection and the ability to discern tumor evolution.
Yeah, so I can only answer that in my highly biased view, but I think there are advantages and disadvantages. So bulk sequencing, it's easier, it's more straightforward. There's a lot of existing data sets, so there's a lot of computational methods that try to deconvolute that data. However, I think at the very best, you can kind of resolve a few of the major populations, but you really can't get into kind of the finer, substructure that's present there. The other issue is that you never really know, 100% which 2 or 3 or 4 mutations are co-occurring in the same clones. You get kind of clusters of mutations, and you can try to estimate that they might belong together, but that you don't, there's no definitive answer on kind of the combination or mutual exclusivity of those mutations. On the other hand, with single-cell sequencing, for a long time, the problem has been we can't really sequence that many cells at a reasonable cost. Or we can't have that much genomic space when we're sequencing for mutations. I think these days we've done a pretty good job at getting the throughput higher to thousands or tens of thousands of cells, so it's not as much of an issue, but there's still technical errors that you need to be aware of in single-cell data. There's a lot of allelic dropout, false negatives, things like that. So they both have advantages and disadvantages, and multi-region sequencing is somewhere in between those two. So.
Great. So we've got a couple of questions. I might bring them to the end to discuss with the entire group, but just one from myself. I was curious, and you sounded curious, why no adipocytes were detected in the last vignette there. Is that a technical concern, question. Or is that,
Yeah, so it was two things. So the first was that, so when you do dissociations, the fat kind of floats to the top, and so you lose a lot of adipocytes there, but not all of them. And the real thing is that we started measuring them under the microscope a little bit, and it turns out the adipocytes are pretty big. They're 100 microns, 200 microns, even larger. So the micro droplets, they have this restriction of size. 50 microns is about the size that you can get them in. And so we didn't get any of them in our cell data, and many other groups doing cell atlas projects also had the same problems. And nuclei ended up being the answer. With nuclei, you dissociate the tissue. The nuclear size of the adipocytes is still about the same as the other cells, and you can measure a lot of the genes that way. So it turns out to be a great way to profile them.
Yeah, that's a nice technical workaround. Great.
Yeah.
Thanks, Nick. So with that, we might press on and invite Vanessa to continue the conversation. So Vanessa, the stage is yours.
Thank you very much. So I'm really here to talk about two of my pet projects in oropharyngeal head and neck cancer and lung cancer to kind of showcase how we might progress single-cell genomics into the cancer clinic. So I'll just start with a bit of an introduction in both of these cancers and then move on to some of the work that I've been doing. So if we look at lung cancer, this is a very challenging cancer to treat in the clinic. It's common and highly lethal, with most patients succumbing to their disease by 2 years. Over the past decade, we've really seen an increase in the use of immunotherapy with checkpoint blockade, which restores T-cell function, and this is being used to improve patient outcomes. Unfortunately, the biomarkers that we have available to us in the clinic perform very poorly. So the best one we have is staining tumour cells for PD-L1 expression. We know that patients who have an expression of above 50% have about a 50% chance of responding to single-agent immune therapy. So that's about as good as flipping a coin. And we're trying to work on methods of how can we improve the predictive value of a biomarker in the clinic to help inform patient care. If we move to oropharyngeal cancer, this is a really interesting cancer where the etiology has completely changed over the last 30 years. This used to be a cancer of patients who heavily smoked and drank alcohol, but now is very commonly associated with infection of human papillomavirus. With this change in etiology, we also have a change in the patient population. These patients tend to be very young. They, as a group, have a superior prognosis to their smoking and drinking counterparts, but we know there is a small group that perform very poorly and have a very high risk of recurrence. The treatment for these cancers can be very toxic and disfiguring. It can include surgery, radiotherapy, chemotherapy, and in such a young patient population, these issues are at the forefront of everyone's mind. At the moment, we really don't know those patients who need a triple therapy approach opposed to a less toxic approach with dual or unimodality therapy. We've had a few high-profile patients over the last few years, which is doing a good job in raising awareness for a very underrepresented group of patients. So if we just start with a case to give you a feel of how this all might work, this is a very typical case, a 78-year-old female who presents to her GP with a troublesome cough. She's found to have metastatic lung cancer. You can see in that PET scan there's a large lung mass there with some bony metastases. She has an adenocarcinoma, one of the most common forms of lung cancer, with a PD-L1 expression of 70%. She also has rheumatoid arthritis requiring intermittent treatment, and so she comes to see me for discussion of treatment. All I can really tell her is that immune therapy may work. She has about a 50% chance of getting benefit from this therapy, but we also know she's probably got about a 30% chance of having a flare of her rheumatoid arthritis. So as you can see, these issues are quite difficult when you're faced with a patient and you really have a lot of uncertainty about the efficacy of therapies. So this is the design of one of the research projects that I'm doing now. We're using single-cell RNA sequencing to assess the response of immunotherapy ex vivo. So patients with advanced lung cancer who are having an endobronchial ultrasound-guided biopsy have a sample taken for research. So we get a fine needle aspirate, usually from an affected lymph node, where we get cancer and immune cells. The cells are then plated in the lab under control conditions or exposed to immune therapy, so either nivolumab or pembrolizumab is what we're using, and then those cells are sequenced separately. And the aim of this project is to see if we can actually measure an immune response mounted by those immune cells in a given patient, which may predict response in the patient themselves. So if we just move on to a couple of the preliminary results, I thought before we could actually describe any change within a patient, we needed to be able to demonstrate that changes in separate patients could be demonstrated. So this is two separate patients, sample A and sample B. They both had a neuroendocrine cancer of the lung. You can see when we've run the sequencing, we get very similar cell subgroups there. But when we look closer at how these patients look side by side, we're starting to see some distinct differences. So this is looking at some site-seq data. You can see in patient B, we have a much more immunoactive phenotype in the monocyte population, as opposed to the immunosuppressive phenotype seen in patient A. And the scatter plot is just a different way of looking at that same data. When we look at the protein expression in the T cells, you can see again that patient A has a population of dysfunctional T cells and activated T regulatory cells, and that the expression of cytotoxic protein in the T cells was different between the two. So I was satisfied that we can actually demonstrate inter-individual differences. When we're looking at the differences in a patient, when we're comparing cells that have been exposed to immunotherapy versus the control cells, we're also starting to see some distinct differences here. So here, the red cells have been exposed to immune therapy, and the blue cells are the control cells. And you can see there that we're seeing some differences in cancer cell death and cancer cell division. So this is still a work in progress. We're trying to work out the very best sort of experimental techniques here. But the hope is that we'll be able to predict ex vivo which patients are most likely to mount an immune response and therefore derive some benefit from the therapies themselves. So I think that it is possible to develop a real-time functional assessment of immune activation to personalize immunotherapy choices in lung cancer. We've also got some other projects looking at single-cell DNA sequencing to look for treatment-resistant clones in the oncogene-driven lung cancers, and whether we can use PBMCs or liquid biopsies to look for treatment response or resistance. There is also some applications in screening which we can discuss later. So if we move right on to the oropharyngeal cancer project, this is a very typical patient, a 49-year-old man, married with 2 children, and is found to have a right tonsillar cancer which has spread to the lymph nodes in the right side of his neck. There's a PET scan there which shows the tonsil cancer in picture A, and in picture B you can see that lymph node on the side of the neck is lighting up. He undergoes surgery, a robotic resection with an oropharyngectomy and a dissection of the lymph nodes in his neck. And one of the lymph nodes shows that the tumour is starting to grow outside the capsule of the lymph node. Now, this is traditionally a marker of poor prognosis and a reason why we would add radiotherapy and chemotherapy to the patient's treatment. However, these markers have been derived from clinical studies that are nearly 20 years old in an era when HPV infection was not very common. So these patients, we really don't understand what constitutes a poor prognosis. And when we're trying to work out what therapies to give the patient, there is a lot of uncertainty. So what we're doing in the lab is using single-cell RNA-seq to assess heterogeneity and aggressiveness in these HPV-associated tumours. So when I talk about heterogeneity, we're talking about genetic variation leading to a variation in phenotype and cell behaviour. So we're taking patients with matched primary and lymph node metastases and using single-cell RNA sequencing. We're using a pooling technique to reduce batch effect and also reduce our sequencing costs, and using a demultiplexing strategy, in this case with Premaxlet. So this allows comparisons between patients, but also between the primary and metastasis, allowing us to study that metastatic niche and what drives metastasis and what cellular identities in the primary might allow us to predict those patients more likely to metastasize and have a poor prognosis. So this is some of the preliminary data. This is all the data sort of pulled together. You can see this is about 51,000 cells. You can see we've got a nice population of immune cells here, and also some distinct populations of epithelial cells. Now, we know that some of these epithelial cells will probably not be cancerous and some of them will be cancerous. So we're using Infer-C and B to look at copy number variation to try and help decide which of these epithelial cells are malignant. And this is going to allow us to study the malignant cell population, the epithelial non-malignant population, which are infected with HPV, and then the non-infected epithelial population. So one of the major criticisms of single-cell sequencing, of course, is that we're removing the spatial context and we're unable to study the cells as they are in vivo. So one of the strategies to circumvent this is to use a spatial technique. We're using the Visium technique at the Garvan Institute. And for those of you who haven't heard of this, you have specially labeled slides where you can place a section of tissue The sections are covered in little tiny spots. These spots contain capture oligonucleotides with spatial barcodes that are unique to that spot. The tissue sections are fixed and permeabilized, and the messenger RNA is released and binds to these capture oligos. That means that the sequencing data can be localized to a given spot on the slide. So then one of the challenges here is that each spot does not just contain a single cell, it contains multiple cells. And so how do we resolve this issue bioinformatically? And one of the strategies that we're using is using an annotated single cell matrix, which I've just shown you, and then using that to try and deconvolute the spots and work out what cells make up any given spot, allowing us then to study relationships between cells. So this is some of our preliminary results. Those slides haven't translated perfectly. So on the top left-hand side, that's what the sections look like. So a section of tissue laying across a little square. The top row there is what the data looks like without deconvolution. So you can see that the resolution's not great. If you're looking for EpCAM cells, they're kind of spread across the slide. But if you look down on the bottom row, that's where we've got deconvolution, where we've actually bioinformatically tried to work out what's in each spot. You can see that probably the EpCAM cells are just on the edge of the tumor specimen there. We've got a few spots of stroma, but that the macrophages are probably sitting just on the rim of that spot of cancer tissue on the right there. This is another patient here. You can see how, again, with the deconvolution, we're getting much better resolution, that probably the majority of this specimen is covered in cancer cells with not much stroma, but some infiltrating immune cells there. So I think we can use this technique to identify malignant cells with aggressive phenotypes that may identify patients who need a more aggressive treatment approach. We can probably use this in the metastatic setting as well, similar to the lung cancer project, to help tailor therapies. And we are doing some work looking at high-risk patients and whether we can use oral swabs to identify patients who might go on to develop cancer. So here are just some of my thoughts on the pathway to clinical translation, and Nick's already spoken about these. Sample collection is going to be a massive problem in this area. The clinical biopsies are very much centered around formalin-fixed tissue, and we would really need to expand that capability for dealing with fresh tissue if we were going to bring this into the clinic. I feel that we also need some automated cell typing so that we've got reproducible pipelines for cell annotation, allowing for comparisons between patients. We also need some clinically relevant timeframes. There's no good reporting these results 2 months down the track when you have a patient who's waiting for treatment. Do we use fresh tissue? Do we freeze tissue so that we can batch patients and make things more cost effective? We need quick processing and sequencing times. And I think we also need to think very carefully about the scientist-clinician interface. How do we report these results? These are very complicated results. We need to be able to report them in a way that clinicians understand and can translate those into decisions for the patient. So thank you to everyone at the Garvin Weissman Centre for Cellular Genomics and St Vincent's Hospital, and for all the people who have funded this research.
Thank you so much, Vanessa. That was really fantastic to see your perspective from the clinic and how you can see the application of these single-cell technologies. I think there's quite a few questions coming through about this HPV link and the change in etiology that you've seen with that cancer. Anthony Beckhouse asks if you've seen a reduction in the HPV-related cancer since the introduction of the vaccine.
Yeah, so the studies showing that there's going to be an improvement in HPV-related disease at the oropharynx are not as sophisticated as those for cervical cancer. So if you think about when the sort of universal vaccination program came into practice in Australia, we're probably still going to have a lag of about 20 years before we really see a reduction, assuming that the vaccine works for oropharyngeal cancer. Also at the moment, you know, we're vaccinating mainly girls and not boys. So I think we hope that it will work, but there's probably still going to be a lag between when we're able to sort of, you know, not have these patients come through the clinic. So I think probably in my lifetime as an oncologist, this is still going to be a big issue for us.
And a technical question on that note, are you actually able to see the HPV infection in the cancer cells that you're studying in the single cell data? Can you detect the transcript or the consequences of infection?
We can see the expression of HPV genes. And what we're looking at as well is doing some long read sequencing to see if we can also look for integration of the virus into the human genome. So at the moment, we're just looking at gene expression, but we will be going that step further.
That sounds really interesting. And perhaps you could also just expand on the scientist-clinician interface. So especially the data reporting from these techniques. What do you see as the main challenges there?
Yeah, so look, it's really interesting because the bulk sequencing techniques are really hitting the clinic now. So where, there are Foundation Medicine and Caris and a few other sort of commercial entities which are offering sequencing to patients. And so we get these often 11-page reports looking at all the genes that have been sequenced and things that might be positive and things that might have absolutely no relevance in the cancer domain or to that patient's cancer. And there's been a number of models kind of gone through the process over the last couple of years where now we really get a tiered report. So highly relevant actionable mutations, mutations of questionable relevance, and mutations perhaps of academic interest, which are followed by a report saying, here are some clinical studies that might help you manage your patient. So, and that might be a panel that only studies 200 genes. You can imagine how difficult a single cell report might be when you're reporting on immune cells, cancer cells, stromal cells. We might have time point analyses, so baseline after treatment. So I think we need to work out, it's probably going to be situation specific and it needs to be tailored to the question being asked by the clinician. It's going to be difficult, I think, very difficult.
Well, perhaps we can come back to that at the end of this session. We'll just have one more question, I think. This one comes from Charlene Naik. He asks about the immune signatures and whether you see any correlation between the CiteSeq data and your results from Visium.
So, interesting question. This project's a little bit unusual in that we did the Visium first and now we're only just processing the single cell data. So the short answer is I don't know. So we've just finished our second run of single cell RNA sequencing. So we're in the process of annotating those cells and then going and deconvoluting the Visium. The results I showed you are brand new. They're brand spanking new. So I'll keep you updated.
Okay, that sounds fantastic. Well, thanks again, Vanessa. And we'll now hand over to Caris, who will give us the final presentation for this episode.
Hi, everyone. I hope you can hear me. So today I'm going to talk about my interest in blood cancer. And I'm looking at the proteins on the inside and outside of the cells in blood cancer using CyTOF, or the mass cytometry technology. So first, by way of introduction, blood cancer affects people of all ages, and in Australia, it's reasonably prevalent. The most challenging thing is that blood cancer still remains an incurable disease. Many of the drugs that treat blood cancer, they do so by killing the cancer cells. For example, these green cells are cancer cells, but unfortunately, drugs that kill cancer cells, they do so imperfectly. So what you get is a few cells that are resistant to the drug treatment, and this is what we call minimal residual disease. And this is a huge problem in terms of having patients relapse and having the cancer come back again. When we're studying sort of how blood cancer responds to drugs, a lot of the traditional methods take what I like to call a fruit smoothie perspective, in that they view the cancer cells all as sort of one single population. So if you have your favorite boost smoothie and it is a berry smoothie and the main flavor is maybe blueberries, that's all you taste when you kind of see that, when you have that fruit smoothie. So it's the whole idea of looking at things as a bulk population, such as what we do, sort of studying cell death in terms of like looking at it from a Western blot. But what I was really interested in doing was like looking at a way of studying blood cancer sort of more like a fruit salad perspective, whereby if you have your fruits, instead of blending it up all into one smoothie, you sort of arrange them according to color and texture. And when you have a taste of your fruit salad, you can taste each individual flavor and appreciate each texture. So sort of looking at cells that respond to drug on a continuum, whereby if you organize the cells, you get cells that are sensitive and cells that are resistant. And because in blood cancer, sort of identifying even what a few cells do is important, I was interested in capturing the networks of millions of individual cells to really understand what happens when blood cancer responds to drug. So the way we chose to do it was to adopt a protein perspective, and we do so using the technology called mass cytometry. Some of you might be quite familiar with this. Quite simply, the way we do it is we have antibodies that are sort of protein tags, and they are labeled with rare heavy metals. And these antibodies can bind to proteins inside the cells and also on the surface of the cells. So we take the cells and we stain them, a cocktail of cells with the different antibodies. We wash off all the probes or antibodies that are unstained, and we run them through a nebulizer that shoots the cells one cell at a time. Cells go into this ion-coupled plasma that's about the temperature of the surface of the sun. And what it does is it ionizes the cells, and what is remaining is these heavy metal tags. Now, when you run it through a quadrupole, then we use something similar to mass spec, so one cell at a time, to be able to detect what protein probes were bound to the cell. What we get then is simply sort of a barcode for each cell, in which each cell we get expression of different protein markers, and we can use that and analyze it and glean information about the cells. So today I wanted to tell you about how I used that technology, specifically about two sort of research studies that I've been interested in. And the first is in the cancer multiple myeloma. So quite simply, our research question is, what drugs can we pair with standard of care therapy? Multiple myeloma, there are a lot of standard of care drugs, and most patients, they go through a treatment where they start with one drug and then they end up with like 5 different drugs and they develop resistance. So that's a huge problem in multiple myeloma. This was our experimental strategy. We took cell lines in this case, and we treated it with a common drug that treats multiple myeloma called dexamethasone. We then labeled the cells, 1 million cells, with different proteins on the inside of the cells. So in my panel, because I was interested in cell death, what we had is cell death proteins, and they're called pro-survival and pro-death. They're sort of switches that determine whether a cell dies or a cell survives. And whether a cell decides to die or survive depends on the balance of expression of these proteins. So we have the cell death kind of proteins shown in blue here, and there are lots of different cell death proteins. And then these proteins also interact with proteins from different pathways. And we can detect sort of functional expression of those pathways by looking at proteins that are phosphorylated or different stages of the proteins. So we coupled all this together in one big panel, and we stained the cells. And lastly, we did analysis on the cells. So let me show you how this looks like. Here is 10,000 single cells expressed on one single diagram, and each sort of circle represents a cluster of cells. So there are 2,000 clusters over here. Now the first feature you'll see on the diagram is it sort of looks like an arrow of different colors. The blue cells represent cells at 0 time point in treatment. The yellow cells represent cells 24 hours of treatment, and the pink represents 72 hours of treatment. From the arrow, there are several features, and the first distinguished feature is the arrowhead. So to understand what the cells in the arrowhead are, we look at the markers of different proteins that we had in our panel. And the arrowhead, the proteins that are expressed, highly expressed, which are shown in red in the head of the arrow as active caspase 3 and cleave PARP. So these are proteins that the cleavage or the activation of it is dependent on whether the cell is going to undergo death. So this arrowhead basically represents an area of where the cells are actively undergoing death. And the tail of the arrow represents viable cells that are alive. So you can see it sort of makes sense in terms of at 72 hours in the pink point of treatment, you have a lot of cells that are undergoing death. So the other feature is on the tail of the arrow, you can see that there's a little island at the end, and this correlates with simply cell cycle. So if you look at these proteins that are involved in cell cycle, the tail component, that little kind of tail in the arrowhead, is an area that highly expresses the protein phosphorhistone 3, and these are cells that are actively dividing or undergoing mitosis. So by viewing sort of this diagram and overlaying the expression of different proteins, you can sort of tell what are the main groups of the cells. So quite simply, what we wanted to know in the study is what distinguishes apoptotic from viable cells. We know that some cells choose to respond to the drug and some don't. So we did a sort of analysis using the learning forest model, and to cut a long story short and an analysis, we were able to rank our protein expression based on the weightage of protein to tell you which proteins were more important in dividing the cells into either dying cells or viable cells. And the top two proteins were phospho-Crab and MCL1. And MCL1 is a pro-survival protein. So at that time when we were doing our study, it was very interesting because there were papers coming out to say that there were small molecule drugs that very selectively target MCL1, and there are several right now that are in clinical trials. So one of the predictions we have is that MCL1, an inhibitor for MCL1, will work cooperatively with DEX in the clinic to be a very potent drug combination. So we decided to test that. And classically, testing that involved applying concentrations of MCL1 and concentrations of DEX drugs to look in a synergy assay to see what the effect is on killing cells. And what you get is plots like that. So you haven't, so if you look at your top left corner where there's a diagram, so you have drug A of different concentration and drug B. And what you get is basically a flat plane if the drugs don't cooperate together. But if the drugs show cooperation, you get sort of a mountain or a hill. But if the drugs are antagonistics, you get a trough. So you can see here when we did this test using MCL1 and dexamethasone, you get a really large hill. So providing an indication that in the cell line that we tested, these two drugs cooperate together very potently. So we decided to then validate this biologically by testing it in xenograft models of multiple myeloma. And here is a xenograft model of multiple myeloma, and you will see that compared to vehicle tested and single dose of each drug, the combination of both drugs are quite potent. We're also able to do that validation in sort of patient samples, whereby we take patient samples, blood samples from multiple myeloma patients, and treat them in a dish with either single agent or both dexamethasone and inhibitor for MCL1. And you can see here that the way we look at this is we express this as a Bliss score. So any score above the dotted line here, above 0 represents potent combinations. And when we look at 10 patients, you can see that majority of the patients respond really well to the combination. Now, what's interesting and what's been highlighted by previous presenters that I've talked today is there's very variable responses in a patient. For example, the orange patient here responds much more strongly to, say, the purple patient over here. And as this kind of combination moves into clinical trials, we're very interested in knowing whether we can use a technology similar to this to understand what's the difference between the orange and the purple patient. So in this study, to sum up, basically what I've shown you is how we can take a simple biological question and use a very reductionist strategy, asking what drugs can combine with standard of care therapy, showed you the example in dexamethasone, and all this initial work and initial modeling was all done in cell lines. Then we can take it and test it in preclinical models and also in ex vivo patient samples. So we're very interested in applying this now to sort of the clinical trials that are going on with the inhibitor of MCL1. So the second case study I wanted to show you is part of a larger study in mantle cell lymphoma. And this case study was focused on a single patient in this trial, and it's asking why the patient developed drug resistance. So here is a brief history of the patient. It was treated with various different agents, and the last agent it was treated with that seemed to be effective is ibrutinib and venetoclax. So venetoclax targets a pro-survival protein called BCL2, and ibrutinib targets the B-cell signaling pathway. with our clinical collaborators, we got a call one day to say that there was a very interesting patient. And typically mantle cell lymphoma is characterized by CD19, CD high expression on the cell surface. Now this patient had somehow relapsed on these different treatments, and it developed a phenotype of CD19 low, CD5 low. And we were asked to, can you just run a CyTOF panel? We don't know what's wrong, to see if you can get any clues to why this patient has suddenly developed drug resistance. So I would really like to think that when we got the patient sample and it was just a tiny bit of blood, we decided to run sort of a very broad panel. So we looked at immune cells, which are surface markers here in gray. And it's because we wanted to know the nature of like the cancer that was developing, because they were not typical CD19, CD5 cells. And then, of course, because the patient was on venetoclax, we looked at the cell death pathway. So venetoclax targets the pro-survival protein called BCL2, but there are alternative pro-survivals that proteins that are present in people. And then because the patient was on ibrutinib, we decided to look at B-cell signaling pathways. So you can see how we decided to tailor the CyTOF panel to really look at kind of the features that might be present in the patient sample. So in order to express the data, I'm using a t-SNE plot, which you'll all be familiar with because we talked about it earlier. So quite simply, it just organizes things in a two-dimensional plane so that we are able to see sort of multiple 43 different dimensions on a two-dimensional plane. So I like to give this example. Imagine that you got lots of images from the internet, for example, about 2,000 images, and you wanted to organize them in a way that makes sense to your mind in two-dimensional. So you put images that are very similar together, close to each other, and images that are dissimilar, far apart. So your flowers are together and your red flowers are together and they are far apart from, say, your flowers on the right-hand corner, and they're very far apart from your top left-hand corner that were your starfish. So we sort of do this together like a fingerprint, if you like, of a patient. So here's how it looked. This was a healthy donor and a patient sample, and it organizes the cells according to the expression of all the surface markers. And here we're showing CD45. That's a marker in all hematopoietic or sort of blood cells. And you notice that the healthy donor has a certain configuration of the cells, and the patient sample only has two distinct islands, one on the top of the plot and one on the far right-hand bottom corner of the plot. So because the population that I've drawn an ellipse around is not present in the healthy donor population, we know that that population is most likely the tumor cells. And you can see that the tumor cells pretty much dominate the whole patient. So as seen by the clinician, when we looked at various markers for main immune subsets, such as B cells, CD19, CD3 cells, for T cells, myeloid cells characterized by CD11C, and monocytes characterized by CD16. You can see that within the ellipse, the color is always blue, denoting a low expression of those markers. This is in contrast to when you look at the healthy blood, where you get sort of islands of red and yellow and orange, which denotes high expression of the markers. So this patient clearly had a very interesting sort of cancer population. But what was interesting is that the patient was on a drug that targets BCL2 called venetoclax. And as you can see in the patient sample, the BCL2 expression was extremely low. Now, we didn't have a comparison, this same mass cytometric comparison of this patient before the patient was on the treatment. But we do have immunohistochemistry. And before the patient was on the treatment in September 2017, you see that when we did immunohistochemistry of the sections, this patient expressed high BCL2. However, when the patient relapsed in April 2018, you see that all the BCL2 high cells were gone, that you only get BCL2 low left. And this kind of low expression of BCL2 correlates with what we see in our CyTOF study. What was interesting, though, is that BCL2 has a related kind of family member called BCL-XL, and the patient instead started expressing higher levels of BCL-XL. So you can see the coloration of the protein expression change from blue to sort of light blue to green. So we took the patient cells in a dish, and we thought that, okay, maybe that means that this patient therefore developed resistance against BCL2 but became sensitive to BCL-XL. There are various inhibitors of BCL-XL that are available, so we decided to test if the patient's cells would respond to an inhibitor of BCL-XL. So this is what we call a kill curve, or a viability curve. And if you see a straight line, it represents that the cells are viable, so they do not respond to the drug. And if you see kind of a line going down, it represents that the patient cells respond to the drug. So in the two lines going down, the one in brown and the one in green, you see that they both have something in common in that they both have an inhibitor for BCL-XL. So it shows that this patient basically developed a resistance to an inhibitor to BCL2 by overexpressing BCL-XL, and it was something that we could sort of pick up with our CyTOF panel. So I would like to kind of finish by showing by this kind of take-home message to say that we've used mass cytometry technology to really resolve the proteins in the cell death machinery at a single cell level. In biological studies, often we use machine learning to identify parameters that control a biological response, such as the example I gave to you, whether cells respond or they resist treatment. I showed you two examples of studies. Study one was really a reductionist kind of biology-driven study where we showed that MCL1 is able to synergize with a standard of care drug in the clinic to kill myeloma cells. And in study two, it was a single study in a patient where we showed that we're able to identify that BCL-XL can overcome BCL2 resistance. I think the power of these studies is saying that if you have the knowledge of the clinical pathway that may be involved in resistance, you can design sort of a protein panel to be able to pick these things up in the clinic. So I just want to thank everybody involved in the study. So I work in the lab of Associate Professor Daniel Gray, and have worked with various different collaborators, initially developed sort of the CyTOF to study cell death together with Gary Nolan's lab in Stanford, where I did my Fulbright, worked with different people from the Walter Eliza Hall Institute, and it's really a multidisciplinary team that comprised of basic biologists, such as Professor David Huang, Andrew Roberts, bioinformaticians, so I work with Professor Terry Speed, also multiple clinicians from different centers across Melbourne to be able to obtain fresh blood samples, including the BCCC, Peter Mac, and Monash, and also the Royal Melbourne Hospital. I work with consumers, patients, and also Andrew Mitchell that operates the CyTOF machine. So thank you for listening, and I'll take questions.
Thanks, Karis. That was amazing. A really nice example of how you can, have a very specific request from a clinician, I guess, in a help us out. We don't know what's going on, and stepping in with some great single cell technology. So I've got a question here from the audience, and please enter in more questions as you go along. This is one that we might start with you, and we might later ask our other speakers to give their take on it. But what has been your experience, building these case studies? They've been really great, powerful examples of clinical utility of the single cell analysis. But what has your experience been like working with clinicians in a real-time sense? I mean, logistical challenges, that's what we're talking about. What's your take?
Yeah, I've really had a good experience with working with clinicians, and I really see it as a partnership. So a lot of times I rely on them to tell me, what is the clinical problem? What is the most important thing we need from this sample? And I think having a clinician that kind of really understands, when I have this problem, this is how I can divide my samples. Maybe I want to do sequencing, maybe I want to do CyTOF. Having the clinician really have sort of an understanding of what each technology can do and how it can contribute to getting answers very quickly for their patient is very valuable. What I've really enjoyed is to be involved in, we have what we call cluster meetings, where they have clinicians and researchers that work, and this is researchers that work on structural biology, researchers that work on sort of different things, come together maybe once a month and just present sort of what they're working at, so people get a feel of, this is what the technology does, or this is what what we need sort of in the clinic, or in my case, I work with beach mimetics, which are drugs that treat cell death. So it's a sort of a very sort of narrow field. And I guess understanding both parties, sort of understanding what each other can bring to the table and what each other can provide has been really good. Sample collection has been really challenging because oftentimes you can't predict when you have a sample. So it's very usual for me to get an email saying, we got a sample in an hour. And to be able to get high quality samples, to have someone there on standby, maybe you have to change your experiments for the day to be able to process that sample. So it's kind of being a cooperation, I guess, developing that pipeline to be able to firstly understand what each other can offer, and secondly, really try to work towards the same goal. That makes sense.
Absolutely. Yeah. And I guess, I mean, we're on the same page from the research point of view. There's a certain duty of care for us to educate for the technology and what the potential can be for the clinician. So they can't necessarily be jack of all trades, but if they are introduced to it or told about it in a nice setting, they can understand the potential, I guess. So we've got a bit of a question that this field of mass cytometry gets a lot, actually. And so Anthony Beckhouse is asking on Twitter whether with single-cell CytSeq and AbSeq and all of that realm of technologies, will that spell the end for mass cytometry? Do you have any comments or opinions or thoughts on that?
Well, we have to wait and see. I mean, at the present moment, and Helen can give you opinion too, CytSeq and AbSeq allows you to detect proteins on the surface of the cell. It doesn't allow you to detect proteins on the inside of the cells. Most of my cyto panels, they're very focused on particular pathways and sort of functional proteins. So a lot of proteins that you can't detect, sort of like phosphorylated forms of the proteins or something else that you can't sort of detect with CytSeq or AbSeq. I mean, you could, I guess, deduce sort of an activation of a pathway based on RNA-seq data or something like that. And kind of one good example actually is BCL itself, the example I just gave you. So if we were to use CytSeq for that same study, I'm not sure whether we would have come up with the answer. And the reason was that there is a change in BCL-XL protein expression, but that change sort of was regulated epigenetically. So if sort of you kind of did protein surface expression and then you did sort of RNA-seq, you probably wouldn't have picked it up. So sometimes if something is like a gene amplification or something, you would pick it up, and sometimes it isn't. So I guess it's kind of like what I said earlier, knowing the limitations of each technology. And at the moment, detecting something on the inside of the cell is not really compatible with CytSeq or AbSeq. So I do think that there's a lot of opportunities for things to merge, but it really depends on like whether we would be able to develop ways of detecting. I mean, for us that look at proteins, we know how important like functional proteins is. So yeah, I don't know if Helen wants to weigh in because you probably have your own thoughts.
Oh, you got it pretty well. That's good. And I would just say the speed of which you could get the answer. And I think that that really plays in nicely into a general discussion we'll have. And I might just take that opportunity to open it up for more general discussion now if Heather, you'd like to take over. But this idea of real time, clinical relevance timeframe, I think was the phrase that Vanessa introduced, and I think that's a really important aspect to this whole integration into a clinical setting. So
yeah, thanks very much, Helen. Happy to open up the discussion now, but perhaps we'll just stick with Caris in the first instance. Nick was describing all the challenges of working with solid cancers in terms of dissociating the cells and making sure that you're able to get a viable cell suspension from a cancer. So, Caris, can you see advantages working with blood cancers, or do blood cancers have their own unique set of challenges in terms of sample preparation?
Well, I guess we have it much easier if you're able to get a vial of blood, and for CyTOF just run, a small amount of blood. But blood cancer also has a microenvironment. For example, if you look at multiple myeloma, the microenvironment really is the bone marrow. And a lot of studies that have taken a look at the microenvironment can show you that cells, multiple myeloma cells that are in the bone marrow are very different from cells that perhaps you would get an aspirate of the bone marrow or even circulating sort of in the system. And that's an area where we need a lot more work to sort of understand it. So I think that kind of resolution of the three-dimensional structure, or even being able to go into the microenvironment, whether it's a bone marrow or for some blood cancers is the lymph nodes, it's really interesting. So in a way, we have access to a particular type of sample, like peripheral blood, quite easily, but that does not necessarily give us the full picture of what's happening in the patient, because I think being able to understand what happens in the lymph nodes or the bone marrow is also really interesting.
Right, thank you. And Nick, I'd like to also pick up on one of the challenges that you mentioned at the end of your presentation. You were referring to the need for established normal reference tissue, and I was hoping you could expand on that, and maybe you could also link it to what Vanessa was saying about the need to report single cell results in a meaningful way to cancer clinicians.
Yeah, sure. I can elaborate a little bit on that. I think the normal references are important because even in normal tissue, for example, you can find cytotoxic T cells. So does that mean that does that mean that there's a risk, or are there subsets of cytotoxic T cells that might be more predictive? Maybe some express different granzymes or different perforins. So yeah, I think it's for any given cell type, like let's say vascular endothelial cells, there are a bunch of different expression programs, and you need to know what that normal diversity is before you go into a tumor and try to claim that the vascular endothelial cells are somehow promoting angiogenesis or causing tumor progression in some way, and so those should be targeted with, say, an angiogenesis inhibitor like bevacizumab or something else. So I think having that normal reference is important. Every organ system is going to be a little bit different. Maybe some of the immune cell references will be common across different organ systems. But we've been interested a lot in the fibroblasts in tumors, the vascular endothelial cells, and also the myeloid cells. And so for us, it's been pretty important to have those references. Also, when you're looking at pre-malignancies, some of those cell types might undergo some pretty early transformations or reprogramming. And understanding, both the vicinity of those cells relative to the tumor margins, as well as how their transcriptional programs are deregulated, especially in early disease, we think is very informative.
Do you ever see any genetic changes in the microenvironment cells, Nick?
Yeah, it's a great question. We've actually been reporting on this, in little subparts of our papers for almost 10 years now. So one interesting thing is, copy number aberrations. If you look at breast cells, or if you even look at many other organ systems, you can find a small population, usually around 3 to 5% of cells, that just have either gained a whole chromosome or lost a whole chromosome. And you might think those are, precursors to tumors or something. Most of them look like evolutionary dead ends, and they probably undergo senescence or apoptosis afterwards. But there is that genetic diversity. Now, there's also growing interest in looking at mutations in normal tissues, and studies that a number of groups are working on right now, trying to understand, both are there early mutations in the lineage, in the development of an individual or an organ that predisposes them, or are there random mutations accumulated in lineages just from replication errors or other sources of damage? And I think it's a very interesting and new area. There is CHIP, which has become, kind of a growing area in studying a lot of the liquid cancers. In the solid tumors so far, there's some germline mutations that are known, but I think the contribution of mutations that occur after the germline, more somatic mutations, that might give rise to drivers like P53 or PIK3CA in breast cancer or others, is really not that well known, and an interesting area. I think it's probably the farthest away from having clinical significance, but at least in the solid tumors. But it's still very, very interesting from a research perspective.
All right. And Vanessa, I also wanted to come back to you, in relation to the science-clinician interface that you mentioned. And I'm just wondering if it would be more practical for single-cell science to be translated into simplified tests, like a biomarker or a test that can run on existing infrastructure in pathology, rather than asking clinicians to understand the complexity of this sort of analysis. What's your thoughts on that?
Yeah, I think that all of us who are working with these new and expensive technologies would love for the sentinel findings to be translatable into, say, immunohistochemistry or flow cytometry. So looking for a marker that could be simply identified using existing techniques. I personally feel that the clinical sphere needs to start embracing fresh tissue biopsies. I mean, we are doing next generation sequencing for actionable mutations already in the clinic. So things like EGFR, ALK rearrangements, those things are done routinely in clinical practice. And we already run into problems with insufficient DNA being able to be extracted from formalin-fixed tissue. And that often requires patients to go back for multiple biopsies, and in extreme cases means that patients cannot have standard of care tests that we need because accessing tissue is so difficult. So I really feel that our sort of pathology colleagues and the hospitals need to start changing how we manage tissue. And that is going to probably take considerable input from, the research institutes, which are often linked with the hospital. And maybe it does need to be a more kind of collaborative approach to banking and storing tissue. Clearly, the hospitals are not going to have space to have lots of freezers going at the same time. And that will probably improve some of the existing tests that we have, but may enable these more kind of bespoke technologies to function in clinical practice.
Okay. Another challenge, an obvious challenge for single-cell analysis is the cost of this. I don't know who wants to comment on this, but is this ever really going to happen, or is it just going to be too expensive? Perhaps we can go back to you, Nick.
Yeah, sure. I don't think cost is going to be the issue at all. The cost continues to drop, and there's lots of good methods now and methods being developed to multiplex samples together. So hash barcodes and various barcoding strategies. So, it's quite possible to combine, reduce costs at least tenfold, maybe even more. So I see the bigger challenge is more being sample compatibility, as well as setting up these logistical, infrastructures. You can imagine that, wouldn't it be great if there was like a centralized, tissue dissociation facility where they could take patient samples and work with pathology and make cell suspensions, and then those suspensions could be used for single cell sequencing, but they could be used for organoids, or they could be used for xenografts, or many other things as well. And, so I think some hospitals are, of course, have become familiar with that a little bit for those other purposes. But with single cell, genomics, it's even more important. I mean, you need to really have the freshest, most viable tissues, at least for the RNA profiling, probably also for the epigenomic profiling. So I see that more as a major challenge, because there's commercialization of these technologies. If you look at the costs, how much they've decreased over the last 10 years or so, it's just been quite tremendous. So I don't think that's going to be the limitation for the clinic. The more challenge is actually, how do we process these samples and set up the kind of infrastructure for that, and can we convince the pathologists to really do this, given all their workloads and all the other things that they have to do on a regular basis. So.
Yeah, I think as I started to think about this, I was encouraged because the pathologists are already handling complex data. I mean, flow cytometry still gives you reports on millions of cells, and there are ways to interpret that data meaningfully in the clinic already. So I think we should try and learn the lessons from putting those methods into the clinic and see if we can also take a similar approach with single-cell sequencing. And we'll have to wrap up this discussion shortly. I just want to thank all the speakers again for giving us such powerful examples of how single-cell sequencing might help patients. But Helen, if I can just ask you, I know you also work with a whole lot of clinical collaborators. where do they see the most immediate impact of this sort of technology for their patients?
That's a great question. So I think if we can get the speed of which we can get answers to them and integrate that into, treatment decisions, I think that would be just the best. So it's the speed and it's the coordination of the samples, as Nick says, getting the right samples to the right people so that you can get the data and be really confident with it. That would be my take-home message.
Are your collaborators also interested in guiding treatment decisions primarily, or are they more looking at early detection or diagnosis?
Yeah, I mean, the cohorts that we've worked with so far have been treatment decisions mostly. But I'd love to work in, bring it forward, and can we predict ahead of time and do that type of early diagnosis? Yeah, absolutely.
And all this discussion also just reminds me of how brilliant these patients are, that they're willing to participate in these very cutting-edge studies and giving us this really powerful data to hopefully benefit many more generations of cancer patients in the future. So we need to thank them as well. But sadly, I'm going to have to wrap up this session now, and I'll just give a huge thanks again to each of our speakers for sharing their experience and their work with us. And thanks again to the sponsors of today's session. And thank you for tuning in. Please complete the survey that will pop up after this episode. We'd love to hear how you're finding this podcast and if there's anything we can do to improve it for you. And don't forget to sign up for next week's episode. Thank you.